Dear students, in today's lecture we are going to learn about somatic embryogenesis and its application. What is somatic embryogenesis? The name itself indicating that somatic embryogenesis, that means formation of the embryo from the somatic cells, not from the zygotic cells, that is not from the gamete. So somatic embryogenesis is a process by which somatic cells or tissues including haploid cells develops and differentiated embryos to regenerate the plants. Coming to the definition of somatic embryogenesis, we can define somatic embryogenesis is a non-sexual development process which produces a bipolar embryo from the somatic tissue, and this definition was given during the year 1978. How the somatic embryogenesis exactly started? During 1958 and 1959, Stewart and Reinert first reported the production of embryos from cell suspensions of the carrots. From thereafter, the somatic embryogenesis is in the continuation process. The somatic embryogenesis has been induced in a variety of explant. Explant means the plant part which is using for production of somatic embryos. So that explants maybe we can use stem, leaf portion, root, hypocotyls, flower bud, seed, embryo, nucellus and endosperm. Any part of the plant can be used for the production of somatic embryos and depends on the plant species. Coming to the types of somatic embryogenesis, there are mainly two types of somatic embryogenesis. One is the direct somatic embryogenesis and the second one is the indirect somatic embryogenesis. Direct somatic embryogenesis means what? It refers to the development of embryo directly from the original explant tissue without the intervening phase, that is callose phase. Callose means undifferentiated mass of cells. Here in case of direct somatic embryogenesis, we don't have the any callous interphase phase. Directly the embryos are formed from the explant. So how this is possible? Because this occurs through pre-embryogenic determined cells, that what we used to call in a short form is the PEDC, where the cells are committed to embryonic development and need only to be released. So where we can find these PEDC cells means they are found in the embryonic tissues, because they are already predetermined to form the embryos, just we are releasing those cells to form the embryos in the culture medium. Example, if we will take such type of PEDC cells, example scutellum of the cereals and ovules and nucellar cells of the polyembryonic citrus varieties, where this from polyembryonic citrus varieties we can obtain the embryo from the nucellar region, and epidermal cells of the hypocotyls in wild carrot. Here we can see the direct somatic embryogenesis in case of Nicotiana tabacum. This picture was taken from the Pathikayam et al. Who published the paper during the year 2013. Here we can see the different stages of the somatic embryogenesis formation. In the picture A, we can see fused globular stage of the embryos. Here this is the fused globular embryo stage, and this is the early heart shape, and C is a heart shape where we can see the formation of two cotyledons, and D early torpedo stage, and E is the torpedo stage where clear formation is there, clear cut difference we can observe, and F is the cotyledonary stage where these two formation of two cotyledons started. These are the various stages we can observe in the somatic embryogenesis, that is direct somatic embryogenesis. Here we are not observing the any callus formation, we have to remember this word in case of direct somatic embryogenesis. Coming to the indirect somatic embryogenesis. Indirect somatic embryogenesis can be defined as the formation of embryos from the callus or cell suspension or from cells or a group of cells or from the somatic embryos. See, here we have to remember these embryos are formed from callus, that is mass of undifferentiated cells, or from the cell suspension, or from a group of cells, or from the somatic embryos, because these indirect somatic embryogenesis, somatic embryos are also formed from the already formed somatic embryos. That term what we used to call, I will explain in the coming slides. Example we will take, if we will see the examples for the indirect somatic embryogenesis where we can observe this phenomena are from the secondary phloem of carrot, inner hypocotyle tissues of the wild carrot, and leaf tissues of the coffee, petunia, and pollen of the rice. Very important phenomena in case of leaf tissues of coffee where we can follow this indirect somatic embryogenesis. So, indirect somatic embryogenesis from which cells we can achieve? Because their PEDC cells we can achieve the direct somatic embryogenesis, that is embryogenically determined cells, but here in case of indirect somatic embryogenesis we are forcibly inducing the cells to form the embryo. So, those cells what we used to call as induced embryogenic determined cells, that is IEDC. Here you can see IEDC means induced embryogenic determined cells. These are embryogenically determined cells, that is pre-embryogenically determined cells where we can get the direct somatic embryogenesis, and this is the induced embryogenic determined cells IEDC where we will get the indirect somatic embryos. So, the somatic embryos regenerating from eggs, plants or callus, they are known as primary somatic embryos. In case of many cases what will happen, embryos will be regenerated from the tissues of the somatic embryos, that is already somatic embryos are formed, from those again we will get the somatic embryos, those are known as secondary somatic embryos. That is why I used the term here, formation of embryos from the somatic embryos. So, those embryos already formed from the somatic embryos are known as secondary somatic embryos. If they are formed directly from the callus or explant, they are known as primary somatic embryos. Here we can see the difference between direct somatic embryogenesis and indirect somatic embryogenesis. In case of direct somatic embryogenesis, we are taking the leaf explant, then shoot regeneration after 6 weeks, then shoot elongation and rooting will happen. And here in case of indirect somatic embryogenesis, there is an intervening phase of callus induction after 3 weeks of the period, and later embryo formation, then shoot regeneration and shoot elongation, rooting will takes place. And here you can see the clear cut difference between indirect somatic embryogenesis and direct somatic embryogenesis. So, ordinarily somatic embryos originating from cells at the surface of the callus or explant, this is already we know. So, embryos formed from in cultures, they what we used to call the term we used to call them as a embryoids or supernumerary embryos or adventive embryos or accessory embryos. Any term we can use for the formation of the embryos from these somatic cells. So, coming to the difference between the somatic embryos and the zygotic embryos, this is the most important. We should know about the what is the clear cut difference between these somatic embryos and the zygotic embryos. So, somatic embryos are formed by sporophytic cells, whereas zygotic embryos are formed by we know that fusion of gametic cells, male gamete and female gamete both will fuse and give rise to the zygotic embryo. But in case of somatic embryo, it is not like that. It is formed by sporophytic cells, that is 2N. And somatic embryos store less amount of the embryo specific reserves, so that is endosperm, they do not have any endosperm. But in case of zygotic embryos, they have the seed storage proteins, carbohydrates, they used to be present. And a distinct suspensor is absent in case of the somatic embryos. Even though if it is present, it may not be functional as in case of seed embryos. In case of seed embryos, what will happen? A well developed suspensor used to be present. So, that suspensor, what will happen? It will anchor the embryo development and helps in absorption of the nutrient material from the endosperm, and it will direct the embryo towards the endosperm to absorb the nutrients from the endosperm. But here there is no very peculiar role for this suspensor, and it used to be non-functional in case of somatic embryos. Coming to the fourth point, embryos have no vascular connections with the cultured explant. But here in case of zygotic embryos, embryos have vascular connections with the explant. that is vascular connections with the explained here means mother cell, mother plant. With mother plant it is having vascular connections through the funiculus. Whereas in fifth point, coming to the somatic embryos, generally they lack a dormant phase and they often show secondary embryogenesis and pluricotyledonary. Already I told secondary embryogenesis, what secondary? Somatic embryos we used to call them, and somatic embryos, they don't have any dormant phase to germinate, they used to germinate immediately. But in case of zygotic embryos, they show secondary embryo, they don't have any secondary embryogenesis and pluricotyledonary nature, but they have the dormant phase. They sometimes in some species they do not germinate immediately. And somatic embryos show high rate of propagation, whereas we can see low rate of propagation in case of zygotic embryos compared to somatic embryos. So now I think you clearly understood about the difference, what is the major difference between the somatic embryos and the zygotic embryos. So now coming to the difference we will see in the pictorial representation form. Here you can see the male gamete and female gamete, which are haploid in nature. They combine to form the zygote, that is diploid in nature. After mitosis it forms the embryo, which will give rise to the new plant. Whereas in case of somatic embryos, that somatic or body cell which is diploid in nature, which will give rise to the callus in case of indirect somatic embryogenesis, and the embryos which are formed from this somatic embryogenesis are known as embryoids, which will give rise to the new plants. Here you can see the different stages the embryo will undergo: zygote, globular, heart or torpedo stage, that is cotyledonary formation stage and cotyledons, and later expansion, maturation, desiccation, tolerance, dormancy, everything will be there in case of zygotic embryos, which will not be observed in case of somatic embryos. Somatic embryos also undergo different phases like globular, heart and plantlet, and finally we will get the plant very easily without any dormancy phase. But in case of zygotic embryo, we have to see all these phases like expansion, maturation, desiccation and dormancy. Finally it will give rise to the germination and growth of the plant. Coming to the different type of embryos, we used to listen different terms related to embryo. So what are those exact terms which are describing the different type of embryos? Zygotic embryos, who gave this classification? Kohlenbach. During the year 1978 he gave this classification of the embryos. So let us see one by one. Zygotic embryos means embryos which are formed by fertilized egg or the zygote, and non-zygotic embryos, that are embryos are formed by cells which other than zygotes, and these non-zygotic embryos, they are classified into somatic embryos, parthenocarpic embryos and androgenic embryos. Somatic embryos, already we know about these somatic embryos, that is they are formed by sporophytic cells in vitro condition. Such somatic embryos arising directly from other embryos or organs are termed adventive embryos. Parthenocarpic embryos means embryos formed by unfertilized egg, they are known as parthenocarpic embryos. Androgenic embryos means embryos which are formed by male gametophyte, that is pollen grains or microscopes, we used to call as the androgenic embryos. And let us see what are the developmental patterns of somatic embryos, how they will show various developmental patterns, that is during the development of somatic embryos. So in the callus cultures, somatic embryogenesis, how it is initiated? With the differentiation of single meristematic cells. So first single meristematic cell, which used to be in a multiplicative phase, then first it will differentiate it to form a callus, and these meristematic cells are distinguishable from surrounding cells because these meristematic cells used to have dense cytoplasm and prominent nuclei. We can see that prominent nuclei. So here what is the difference between these meristematic cells which are going to form a callus? Means it will be very dense in cytoplasm and prominent nuclei, and this is the difference from the surrounding cells. And further divisions of these meristematic cells, which results in the formation of group of cells, and each proembryoid cell that possess the capacity to pass through the some stages of the embryo development. That is, every pro-embryoid cell have the capacity to undergo these stages like globular, heart, torpedo and cotyledonary, as usual observed in zygotic development. What we are observing in zygote development, same thing here also we can observe. And somatic embryos are bipolar structures. They have both radical and plumule, and radicular end that always orient towards the center of the callus or cell masses, and whereas plumular end always stick out from the cell mass, because plumular end it will grow upward direction, whereas radicular end that will stick to the center of the callus and it will go downward direction. So in case of sometimes what will happen in many cases of somatic embryogenesis, what will happen, radicle used to be suppressed. In such cases we have to initiate the radical formation. And somatic embryos can be germinated readily on nutrient medium without any hormones until the plantlets reach a suitable size for transfer to soil. So after the plantlet formation, then we will give a hardening phase, then we will transfer to the field. So here you can see the different phases. Initial single cell, where this meristematic cells forms with a dense cytoplasm and a prominent nuclei. Then on the first medium, then secondary medium, how it will form different stages like globular stage, heart stage, torpedo stage, and finally leads to the plant formation. Then this somatic embryogenesis entire process can be divided into different stages for our easy purpose of understanding. That is, first phase is the callus initiation, also known as inductive phase, that is we are inducing the embryo formation here. Then second one is the embryo development, and third one is the embryo maturation, also known as expressive phase, and fourth one is the plantlet formation. So first phase, that is inductive phase, the name itself indicating that we are inducing the cells to form the embryos. So here in inductive phase, the somatic cells, they will acquire the embryonic competence and start proliferating to form the embryo. So auxin, particularly 2,4-D, that is generally required to induce the embryogenesis in the initial stages. Then after addition of this 2,4-D, then they will form the pro-embryogenic mass. Then for callus induction, generally which media we will use? MS media, that is Mura-Scheidt-Skog media, that is supplemented with different concentrations of auxins 2,4-D, and this concentration will depend on species, which species you are going to use for the production of somatic embryogenesis, and explant also these concentrations will vary. Then coming to the second one is the expansive phase. Expansive phase involves both development and maturation. In expansive phase, especially in case of developmental phase, we have to transfer to the another medium which is having low concentration of the auxin or without any auxin concentration. Then what will happen, that cells will start developing into a mature embryos. Initially we initiated the differentiation of these meristematic cells into a cells with the help of the auxin, but later during the development we are removing the auxin, or we are providing the less concentration of the auxin to induce the maturation of the embryos. Then these mature embryos will start showing different phases like globular structure, heart shaped structure, torpedo shaped structure. Then after that, during the maturation phase, cotyledonary phase we can see, and followed by germination of the somatic embryos, because they show the high rate of propagation without any dormancy period. Okay. Here also you can see the what are the different phases we can see, that is globular stage, heart stage, torpedo shaped stage, and cotyledonary stage. Then coming to the different factors, they can capable of influencing this somatic embryogenesis. Of course, any process will be influenced by many factors. Similarly, the somatic embryogenesis will also be influenced by different factors. The first one is the genotype and type of explant, that is which type of explant we are going to use for the somatic embryogenesis. Even though a variety of explants we can utilize for inducing the somatic embryogenesis, the correct developmental stage of the explant, that is too much crucial for initiation of this embryogenic callus. Young or juvenile explants produce more somatic embryos than older explants, of course, because young or juvenile explants are very vigorous in multiplication. They are compared to the older explants, hence it is better to go for young or juvenile explants. And different explant tissues from the same mother plant also having different capabilities to produce the embryogenic callus. In case of, if we will take the example in case of Azadirachta indica, that is neem tree, the immature zygotic embryo at a different stages of the development like globular, early to late heart shape, torpedo shape and early dicotyledonous stage, when culture showed varied potential for somatic embryos. So this is direct embryogenesis. In case of neem, here what we are showing, that is direct embryogenesis, which we are taking that is pro embryogenic cells. So they have different capabilities, means they are show different frequencies. Some cells will induce more somatic embryogenesis, some cells will induce low somatic embryogenesis. And explant genotype also has a marked influence on somatic embryo regeneration. For example, if we will take the species like carrot and alfalfa, almost any and every explant show the embryogenic potential, that is any part of the plant from the root to the shoot, any plant part we can utilize them as a explants, and they have equal potential to induce the somatic embryos. But in case of many other species, embryogenic potential is confined to embryonal or highly juvenile tissue, that is younger tissues. In case of cereals, if we will take wheat and legumes, conifers, where immature zygotic embryos have to be used for consistent and high frequency response. These are the different examples. And coming to the second one, growth regulators. First we saw the nature of the explant, how they will influence the somatic embryogenesis, and second one is the growth regulators like auxins. Auxin alone we can use or in combination with the cytokinin, because both are required for the somatic embryos. These are essential for onset of growth and induction of embryogenesis, because cytokinin will promote the division of cells. Auxin will induce the formation of embryos. Of all the auxins, generally we will use the 2,4-D is the most prominently used one, followed by NAA. And the auxin, what it will cause the de-differentiation of the proportion of the cells of the explant, then they will start dividing. Then in the presence of auxin, the embryogenic clumps that grow and break up into smaller cell masses, that is the callus. Okay, then when auxin is either removed or reduced, that is during the maturation phase, the cell density used to be lowered, that is multiplication used to be lowered, then embryogenic callus, which give rise to several somatic embryos, and they believe to develop from a single superficial cell. Of course, we are thinking that they are developing from a single cell. Initially that single cell will multiply and form the callus. From that callus, somatic embryos are formed. And ability to regenerate the somatic embryos, that ability is known as, the peculiar word what we used to use is totipotency. That is ability to regenerate the somatic embryos, that ability is known as totipotency, that is acquired by the cells during this de-differentiation in response to high auxin treatment. Suppose if you will see in case of carrot, coffee, alfalfa, in this species totipotency because of high auxin treatment. So the somatic embryogenesis is a two-step process. First one is the induction, occurs at high auxin concentration, whereas development, which is occurs at low auxin concentration, or growth regulator free medium, that is development and maturation. So totally it is two phase, induction, then development and maturation, both combined to call as expensive phase. Expensive phase means it is consuming everything, we are not using any growth regulators, that is the expensive phase. And in the somatic embryo induction phase, explant cells differentiate and become totipotent. When embryogenic callus are transferred from induction medium to an appropriate medium, Then somatic embryos differentiation proceeds from different stages like globular, heart-shaped, torpedo to cotyledonary stages, and this is known as somatic embryo developmental phase. And then another growth regulator is the cytokinins, and cytokinins have been used in the primary medium invariably during the embryogenesis of croplands. And what is the effective concentration of cytokinin? That is from the 0.5 to 50 micromolars. And cytokinins, what they will do, they are important in fastening the somatic embryo maturation and especially cotyledonary development. So growth regulators also, and another one ABA, abscisic acid. So why abscisic acid has to be added for the formation of somatic embryogenesis? Because we used to add at inhibitory levels of 0.1 to 1 micromolar, which will promote the somatic embryo development and maturation. At the same time, it will inhibit the abnormal proliferation and initiation of accessory embryos. Then coming to the third factor, that is the nitrogen source. So nitrogen, which form we have to give? Nitrogen source has marked influence on somatic embryogenesis, especially in case of carrot. Ammonia form has a promotive effect on somatic embryo induction, while development occurs on a medium containing nitrate as a sole nitrogen source. So induction will happen on the ammonia form of the nitrogen, whereas expansive phase occurs on the nitrate phase. In case of alpha-alpha, there is absolute requirement of ammonia during induction plus differentiation. In both phases, ammonia form is required. So it depends on crop, which crop we are using for somatic embryo formation. Like other factors like high potassium levels and low dissolved oxygen, these will also influence the somatic embryo regeneration. And in case of citrus medica, some volatile compounds like ethanol, they will inhibit the somatic embryo regeneration. In case of soybean, low sucrose concentration, it will promote the somatic embryo regeneration as compared to high concentration. These are already standardized, like researchers, they used to standardize how much concentration is exactly required for the somatic embryo formation in different crop species. In case of alpha-alpha, use of maltose as a carbon source, which will improve the both the somatic embryo induction as well as maturation as compared to on the sucrose medium. Because energy source generally we will use the sucrose, but in case of alpha-alpha, maltose has the more influence. And somatic embryo germination media like MS media we will use. In that we will use the BAP and NAA with different concentrations, and temperature we will keep at room temperature, that is 25 degrees centigrade plus or minus 2 degrees centigrade with a 16 hours light, and this light will be provided by the fluorescent bulbs, because these experiments has to be conducted under decontaminated conditions, so there should not be any contamination. And calculations how we will do for somatic embryo formation means callus induction frequency is how much and regeneration frequency. Okay. Then what are the different applications of somatic embryogenesis? Up to now we saw that what are the different type of somatic embryogenesis, direct and indirect, what are the different factors influencing the somatic embryogenesis. Now let us see what are the different applications of somatic embryogenesis. So first application is somatic embryogenesis, that is somatic embryos, they may replace the micropropagation technique for rapid propagation of economically important plants, so that is commercial crops. In case of commercial crops, if somatic embryogenesis is standardized, then we can use that technology in the place of micropropagation. Then somatic embryos can meet specific breeding objectives by rapidly multiplying the germplasm that is initially present as embryogenic material. And like example, maternal embryos, haploid embryos and interspecific hybrid embryos, which normally generally abort due to non-availability of endosperm tissue. This I explained in the last class about the embryo rescue technique, embryo abortion. If you need, just I will provide that link in the description box, just you go and watch that video. Then rising the somoclonal variations from tree species, especially in case of indirect somatic embryogenesis, we can observe the somoclonal variations. And synthesis of artificially synthetic seeds and it is a regenerable protoplast system also, the somatic embryos. And in case of citrus, we will use the nucellus for the regeneration of the somatic embryos. That is, nucellus usually degenerate during the development of seeds, but in case of citrus species, embryogenic callus, which is derived from the nucellus, remains totipotent for many years. That is very advantageous in case of citrus. And non-chemical mutants, generally to obtain the mutants in the breeding program, we will apply the chemicals. So here, because of somatic embryogenesis, without using the any chemicals, we can obtain the mutants. How? Means through adventive embryogenesis in case of tissue culture experiments. Then through somatic embryogenesis, main advantage is we can obtain the disease free plants using the nucellar embryos. Why disease free plants? Means any pathogen, it cannot reach the embryo stage, because embryos are actively dividing, very active phase. If we could capable of isolating the cells which are do not infected with any pathogen, then we can obtain the disease free plants. This is the case different from the seed-borne pathogens, that is the different case. But here I am saying that we can obtain the disease free plants by using the nucellar embryos. In citrus especially, citrus is infected with a lot of viruses. So in citrus, certain viruses which infect the vegetative tissue, they can be eliminated from the nucellar cells during the ovule development. So somatic embryos which are developed from these nucellar cells, they produce the rejuvenated clones which are free from the viruses. And there is also a possibility of chimeric embryos which are arising from transformed and non-transformed tissues of the callus. They can be bypassed through the process of repetitive somatic embryogenesis. So here we can avoid the somatic embryos also. And repetitive embryogenesis is of potential use in case of synthesis of metabolites such as pharmaceuticals and oils. That is also very advantage. However, each technique have its own limitations. Similarly, somatic embryogenesis have its limitations. What are those limitations? Let us see one by one. In many species, somatic embryo maturation and conversion remain problematic, and resolution of this bottleneck is critical to the practical utilization of this somatic embryogenesis, because maturation and conversion into a plantlet is a very difficult task in case of somatic embryogenesis. First we have to overcome those problems, then only we can utilize the somatic embryogenesis on a commercial scale. And occurrence of somaclonal variations in case of indirect somatic embryogenesis, already I told in. And somatic embryos are without seed coat. Of course, they are the embryos, they don't have any seed coat, because as we observed in the zygotic embryos. So what we can do is we have to cover the somatic embryos with artificial seed coat, that is sodium alginate is the material what we can use for the formation of synthetic seeds. That sodium alginate, it will act as a protective gel to protect these somatic embryos during the transportation. And unfavorable conditions also, to protect the somatic embryos, we can use encapsulate material, that is the sodium alginate. And abnormalities exhibited by somatic embryos, which include like double and triple vascular systems, so secondary embryogenesis and pluricotyledonary, these are disadvantages only. And large scale production is also very difficult, already I told. And quality is also often poor compared to zygotic embryos. Of course, zygotic embryo is properly developed, but somatic embryo artificially we are inducing, so obviously the quality of the somatic embryos will be poor only. So we have to improve the techniques for improving the quality of the somatic embryos. Then field conversion percentage is also, that frequency is also less in case of somatic embryos and artificial seeds. That is around, if we are capable of producing 100 somatic embryos, only 15 to 20 we can capable of converting into a new plantlet. So that frequency is also less. And synchronization of somatic embryogenesis is also inadequate. That is, all the somatic embryos, they should be in the similar stage, but here synchronization is very difficult. Some cells are in heart shape, some cells are in torpedo shape, so it will be difficult. So that synchronization also have to be achieved before commercialization. So let us see some of the questions regarding the somatic embryogenesis. First one is the primary hormones required for differentiation of callus are first option auxin and abscisic acid, no. Second one is the ethylene and auxin, no. Third one is the cytokinin and gibberellin, no, because only we need here cytokinin is okay but gibberellin is not. Then fourth one is the auxin and cytokinin. Yes, these two are the main hormones we will use in the somatic embryogenesis process. Which of the following material will be used to encapsulate somatic embryos to produce the synthetic seeds? Already I told sodium acetate, no. Then sodium nitrate, no. Sodium chloride, no. Answer will be the sodium alginate. This will be used as a encapsulate material for the production of synthetic seeds. And third question, in plant tissue culture, the complete plantlets can be generated from callus tissue by altering the concentration of sugars, no. Hormones, yes. Amino acids, no. Vitamins and minerals. Why means hormones we are altering, we are reducing the auxin concentration at the maturation phase. Okay, so answer will be hormones. Then what is the callus in somatic embryogenesis? Callus means first option is tissues that grow and form an embryoid, no. An unorganized actively dividing mass of cells in a culture, yes, it is the right answer, because callus is a unorganized actively dividing mass of cells in a culture. Then C option, an insoluble carbohydrate, no. Then fourth, a tissue that grows from an embryo, no. Only option is an unorganized actively dividing mass of cells in a culture. So thank you students, this is the lecture for today's class. I hope so you will enjoy this lecture, and I think it will be very useful for you. If you feel that it is very useful, then please share with your friends and subscribe to my channel to upload the more videos, and don't forget to subscribe for my channel, and also suggest to your friends regarding this channel. And thank you for your keen interest in this video, and remember to like and subscribe for more videos. I wish you a great success, health, prosperity and happiness. Let us meet with the next video. If you have any doubts regarding this lecture, please post in the comment box. I will answer those questions. Thank you one and all. Bye bye.